Today is the fifth and final day of our prestigious GIAN course on Advances in Experimental Therapeutics, and we are thrilled to begin this conclusive day with the same energy and academic zeal we have maintained throughout the week. This course has been a significant platform for knowledge exchange, research insights, and international collaboration. On behalf of the entire organizing team, I extend my heartful thanks to one of you, our esteemed speakers, coordinators, and participants for making this event a resounding success. Your efforts, vision, and guidance have shaped this course a truly impactful learning experience. To begin today's academic proceedings, we invite our distinguished international expert, Professor Sanjay V. Malhotra, to deliver the lecture on the case studies.
No, I don't watch.
No, you have to click on duplicate.
I will, यहाँ पे ना, I'll go there now. Thank you. Thank you, Tanya. Good morning, everyone. So what I thought probably I will share with you is now I showed you a few examples of, as I was telling, experimental therapeutics. Hopefully, with what I have shared with you, you realize the fact that it's a multidisciplinary enterprise. It's a multidisciplinary field where many disciplines, many things come together to develop a therapeutics, even to understand how the therapeutics works. So what I thought now in the final, this lecture is to bring all of these things together to show a case study where you will see the applications of many of these utilities, many of the modalities, applications that I have been telling individually, from imaging to therapeutics to analysis to PK and all of that, how these are actually employed, used for developing new therapeutics. So my lab, my work, as I showed you, as I told you, I'm a trained chemist. Synthetic organic chemist. I did my bachelor's in chemistry, pure chemistry, and then applied chemistry, and then again synthetic chemistry, catalytic chemistry. So my training has been purely on chemistry. But as I mentioned, as a chemist, I had the curiosity, and fortunately, the opportunities to be picking up those projects which taught me a lot. Because not that you enter into project and you सब कुछ आपको आता ही है, सर्व संपूर्ण कोई नहीं है. So I started as a chemist. The reason I'm sharing with you is how this journey began and what we do. So now in my lab, for example, what we focus on is in the area which is in the black. So top bar पे जो आप देख रहे हैं, it is the process, the path जो drug discovery, drug development. So यहाँ से target identification is something that, for example, for a disease you have a target. Target could be initially what disease type is. Let's say you're interested in a type of cancer or neurodegeneration. In there, then you want to understand what the biological target is. So biologists working with you will say, okay, हमने जो study की है, this seems to be the target. Sometimes they do, sometimes they don't. Presume based on the data that they have. Once they do that, my lab also does that, but once they do that, after that it becomes whole experimental therapeutics part. To identify which modality will work, how it will work, small molecules will work. So I have written here small molecule, but that's not the only thing we do. We do peptides, antibody drug conjugates, mostly these modalities that we work on. So this path that shows is you are do the high. Once you have identified what your target is, you develop an assay. As I was telling you कि assay होना चाहिए कि identify करने के लिए कि protein को आपने follow करना है. You develop an assay, and then you screen compounds, whether they are known compounds or unknown compounds. You screen thousands of compounds, you design compounds, and I'm going to share with you how we designed the compound, from where we started based on the knowledge of that. And then you again lead optimization. एक बार आपने hit निकल आया, उसको optimize करना lead, कि अब उसको जो है ना आपको यहाँ, remember I showed you arrow टारगेट में दूर है उसको लेके आपने बुल्स आई एकदम सेंटर में लेके आना है टारगेट पे. दैट इज़ द ऑप्टिमाइजेशन प्रोसेस. देन लीड प्रोफाइलिंग, सारा एडमी डॉक्स. अब उसका प्रोफाइल आपने चेंज करना है कि इट इज़ सेफ and efficacious and something that is really going to be helpful. Due to the lead profiling, then it enters the clinical part, जो कि मैं yesterday, as I told you. So my lab, our center works on all of these things, and we employ many different techniques. So we design molecules, we use chemical biology to understand the biology, the tools that we create. These tools are the molecules that you create, generate, use to study biology are called chemical tools or probes. They are probing the biology. We use genetics. Genetic level पे उसकी क्या information है to study that. We design de novo scaffolds, new compounds. We take motivation from what we learn from nature या जो published में है, but then we see if that is helpful. Otherwise, we design as a synthetic chemist, medicinal chemist. We design our own modification करके कि how it will help us to understand the biology of the disease. We do a lot of imaging, and as I told you that we have a whole cyclotron and we have a whole radio pharmaceutical synthesis enterprise. We do a lot of imaging के लिए design molecules so that you can image the in vivo studies and make sense of that. We study radiation because radiation, when you radiate, it's a DNA damage when you do this. So when you do the DNA damage of a target in a cell, whatever, let's say cancer cell है उसके अंदर if you're doing, what happened? How do you manipulate the radiation in combination with the drug? So we do a lot of those studies. And then in this process, if we identify a hit compound or compound which is really helpful, then we jump on this path. जो नीचे है सारा, this is what the pharma industry does. कि आपने एक ड्रग लिया, जो लीड को डिस्कवर किया, you discover, and then you optimize the lead and you keep on iterative process जब तक कि जैसे as I was telling you, उसकी properties क्या हैं, admitox, solubility, bioavailability, non-toxic safety. You keep on studying these things and optimizing. उसको आप पॉलिश करते रहते हैं, उसको एडिट करते रहते हैं, उसके एनालॉग्स बनाते रहते हैं. सो वी डू ऑल ऑफ दीज थिंग्स. और एक बार सक्सेसफुल हो गया, then it enters the drug development stage. So we do most of this in-house in our center, in our institute, in my lab. So when we do this, we employ many of these techniques. As you see here, we pharmacology, genomics, imaging, and immunology. We study discovery से लेके preclinicals, IND enabling से वहाँ थे. We use bioinformatics, biostats, biomarker development, and of course we are educational. We are an educational institute, so education is part of whatever we do. And as you see here, before it enters clinical phase, right from the beginning we are interacting with all these experts area. We do a lot of this in my lab, but as and when needed we engage. So it's a collaborative enterprise. No one person can easily may have वो तो उससे अच्छा तो हाथ रखे के तथास्तु और हो जाएगा काम. इसमें काम करना पड़ता है. So this is a multidisciplinary enterprise that we do together. So what I'm going to share with you now, an example of how in my lab our group started employing many of these techniques and different pieces जो मैं अब तक आपको 4 दिन से बताता आ रहा हूँ, हमने सबको कैसे मिला के एक लीड कैंडिडेट को कैसे बनाया. So you'll see a single candidate निकालने के लिए किस तरह से सारी जितनी भी जो एक्सपर्ट जो डिसिप्लिन्स हैं, नॉलेज हैं, it comes together to do that. Now what is cancer? So we know that cells divide, and when they divide they accumulate DNA damage. DNA का डैमेज होता है क्योंकि ब्रेक होता है, डिवाइड होता है, रिपेयर होता है, एंड मोस्ट ऑफ द डैमेज इज रिपेयर्ड. कुदरत ने बनाया इस तरह से है कि द डैमेज गेट्स रिपेयर्ड. बट इफ इट इज नॉट रिपेयर्ड, सो बेसिकली क्या है, you know the chemical reaction. I thought there was a knock on the door. So if it is not repaired, it's a basically chemical reaction, bond breaking, bond making. So there is a kinetics involved. आप अगर physical chemistry अगर आप याद करें तो उसका जो है there is a rate of reaction. Bond break होता है, bond बनता है, bond break होता है, bond बनता है. It happens in picosecond speed से. And if for whatever reason, so and most of the time these things are repaired. Occasionally there is a bond जहाँ से, as I was telling you, DNA what is many base pair, many base pair, amino acid, nucleic acid, carbohydrate, fatty acid. I showed you polypolymer. वो base pair हैं जो connect करके प्रोटीन्स बनाते हैं, आरएनए बनाते हैं, डीएनए बनाते हैं. सो कुदरत की रचना ऐसी है कि इट हैपन्स एग्जैक्टली एट द सेम प्लेस अगेन अगेन, राइट. सेल्स इसी से क्रिएट होते हैं. अगर नहीं हुए, इट अटैच समवेयर एल्स, इट बिकम्स, सो जहाँ से बॉन्ड बनना चाहिए अगर वहीं जाके नहीं लगा, 1 in 10,000 टाइम्स. जस्ट इमेजिन, अराउंड 1 in 10,000 टाइम्स. आप यहाँ बैठे हैं, हम सब यहाँ हैं इस रूम में, हर एक के सेल्स के अंदर वो डीएनए जो है ब्रेकिंग मेकिंग चल रहा है, सेल फॉर्मेशन इज गोइंग ऑन. सो यूजुअली जो एज पर द स्टडी शो के अराउंड 1 in 10,000 टाइम्स वो बॉन्ड सीधा वही नहीं लगता है. इट्स अ केमिकल रिएक्शन. आपने देखा अमीनो एसिड का स्ट्रक्चर क्या है, इट फॉर्म्स समवेयर एल्स. एक्सक्यूज मी. व्हेन दैट हैपेंस, वो अब उसने थोड़ा स्ट्रक्चर चेंज हो गया, इट्स कॉल्ड म्यूटेशन. वो म्यूटेट कर गया, ठीक है. सो दीज mutations can occur anywhere on the DNA, right? DNA इतना बड़ा है, सारे functional groups लगे हुए हैं, amino acids वगैरह. So that mostly and जो mutations इकट्ठे होते हैं, these mutations are called passenger mutations. वो साथ में चलते रहते हैं. आपका सेल डिवीजन हो रहा है, वो साथ में चलते जा रहे हैं. there isn't much really happening. they are harmless. आपके सेल के अंदर वो साथ में चलते जा रहे हैं. occasionally what happens is these mutations become important and really of interest to the cell. सेल की ग्रोथ के लिए वो इम्पॉर्टेंट होता है. सेल फास्ट ग्रो करने लगता है. नाउ व्हेन इट स्टार्ट्स ग्रोइंग फास्ट, इट स्टार्ट्स टू दोज म्यूटेशंस, धीरे धीरे वो कोडॉन्स जो है इकट्ठे होने शुरू हो जाते हैं. एंड स्लोली दैट ग्रोथ बिकम्स फास्टर. नब कुदरत ने बनाया है आपका इम्यून सिस्टम, आपका फाइटिंग मैकेनिज्म जो है इंसान की बॉडी में, बायो क्रिएशन बॉडी में, वो इम्यून सिस्टम आइडेंटिफाइज एंड ट्राइज टू क्लियर इट अवे. वो एक्सक्रीट कर जाता है, बाहर निकल जाता है, क्योंकि इम्यून सिस्टम रिकॉग्नाइज दिस इज समथिंग फॉरेन एंटिटी, वो उसको बाहर कर देता है. बट समटाइम्स दे आर सो रिजिड, क्यों बाहर नहीं होते, दे कीप ऑन collecting, and this becomes a lesion. And but sometimes these lesion again evades the immune system को. immune system को वो धोखा देके आगे निकल जाते हैं and eventually grow, and it's called cancer. क्योंकि आपको पता नहीं है कि म्यूटेशन क्या हो गया, कहाँ से हो गया. the cell is growing faster. It's not the normal cell. Now it's growing faster. Immune system को beat करके वो आगे निकल गए. So that is grown. Now what happens is identified. Patient goes to the doctor. Doctor looks at, identifies कि किसी एक जगह पे उन्होंने लीजन उन्होंने नोट किया, डायग्नोस किया. डॉक्टर के टूल बॉक्स में जो भी दवाइयाँ हैं, जो भी अप्रोचेस हैं, वेदर इज अ सर्जिकल अप्रोच है, रेडिएशन अप्रोच है, कीमोथेरेपी है, इम्यून थेरेपी है, जो भी अप्रोचेस हैं उनका कॉम्बिनेशन है, देने की कोशिश करते हैं. एंड देन व्हाट हैपन्स इमीडिएटली, उस समय जो अटैक होता है कि दोज कैंसर सेल्स डाई, निकल जाते हैं, और जो डायग्नोस होता है उसमें नज़र नहीं आते. बट उसमें कुछ टाइम बाद में फिर से पेशेंट जाता है. नाउ डॉक्टर सीज कि it's not at the same place, but now it has spread it in other parts of the body. Because you cannot diagnose, but if the cell थोड़ा सा भी रह गया है, they grow fast. वो grow करके, and then doctor will give another, whatever is available in his or her kitty toolbox. Again, it suppress किया, and after some time, initially it's suppressed, and then it comes back. You try again, doctor tries again. Eventually, after some time, it becomes the race. क्योंकि जितना डॉक्टर के उसमें जो तरीके थे, सर्जिकल था, रेडिएशन था, कीमो था, इम्यून था, जो गया वो कॉम्बिनेशन सब कुछ ट्राई कर चुका, but it's still evades. And that's when you hear the term कि दवाई काम नहीं कर रही, right? ये I'm trying to tell you as a scientist, समझने की कोशिश कीजिए कि दवाई काम नहीं करी उसका मतलब क्या है. Means this कि जो दवाइयाँ दी थी, जिस चीज़ के लिए दी थी, right, उसके बावजूद वो evade करके grow करा है. So and what say कि patient has developed resistance to the treatment, right? These are the terminology, common term used. Scientifically क्या हो रहा है? जो drugs बनाए गए थे, remember now you connect this to in past 2-3 days what I've been saying. Drug अगर बनाया है उस टारगेट के लिए नहीं बनाया है तो शुरुआत में तो इतना कीमो इतना स्ट्रांग होता देंगे तो कुछ दिन के लिए सेल वाले को मार देगा क्योंकि इट्स वेरी है, बट उस टारगेट को नहीं दबाया है तो कहीं से भी निकल कर नजर नहीं आता, फिर से निकलना शुरू हो जाएगा. सो देयरफॉर डिजाइनिंग अ ड्रग विच इज स्पेसिफिकली विद दैट प्रोटीन, नाउ यू सी कि हाउ इट्स इंपॉर्टेंट. अदरवाइज वो इनिशियली तो असर होगा क्योंकि अब इतना टॉक्सिक चीज़ दे रहे हैं. दीज आर हाईली टॉक्सिक केमिकल्स and they initially they are bombarding and there are side effects that you see unfortunately. The patient goes through a lot, body loss, weight loss, hair loss, all that, क्योंकि वो टॉक्सिसिटी है ना, उसका साइड इफेक्ट है. बट उसके बावजूद भी जो ड्रग दिया गया है, जिस टारगेट जो है उसके लिए अगर नहीं बना है तो पेशेंट गोज थ्रू ऑल दिस ट्रबल, ऑल चीज़ के लिए कुछ टाइम के लिए सप्रेस हो जाता है, बट टारगेट के लिए तो बना नहीं है ना, टारगेट उसके लिए वो अटैक हो नहीं रहा है. सो इट कम्स बैक and we call this resistance. Now over the years have been made to understand what is resistance, ऐसा क्यों होता है, and there could be many different reasons, and these are the few primary mechanism. It could be कि प्रोटीन जो है उसका फंक्शन उसने अपने आप को बदल लिया है. So नेचर इज प्रोटीन जो दे आर वेरी क्रिएटिव. आपने जो दिया लगाया उसको थोड़ा मॉडिफाई, हल्का सा मॉडिफाई कर गया, अब टारगेट के ऊपर drug is not working. Could be one reason. It could be, for example, efflux. This is apne, I don't know, because you're not biologist, you probably know. Do you know what is PGP pump? Okay, so PG pump is a protein जिसके कहते हैं that pump efflux. जो pump है वो जो proteins हैं, they identify some foreign entity, they throw it out. तो drug आता है, वो drug को बाहर फेंक देते हैं. So this PGP pump efflux होता है बाहर बन के, तो ड्रग पहुंचा नहीं टारगेट तक. That could be another mechanism. There could be, there is passive diffusion. There could be there is a DNA damage हो गया है, it's not prepared. So there are 6 variation. ड्रग एक्टिवेशन अंदर जाने के बाद में ड्रग का स्लाइटली मॉडिफाई हो गया, मेटाबोलाइट बन गया, अंदर के सेल एंजाइम के कारण इट्स नॉट वर्किंग. So there are 6 at least reasons which have been identified that you see I have mentioned here: epigenetic reflux, DNA damage, cell death, EMT, epithelial to mesenchymal translation, which means the drug goes from the cell of site to away from there. It is EMT, MET coming back into. So drug target alter हो गया है, change हो गया, drug is becoming inactive. So there could be many, and there are encyclopedic information available on each one of these, but there could be many reasons for which it's not working. So in my lab we started to study looking at paclitaxel. I think I was telling yesterday about one third क्यों काम करते हैं करके. Paclitaxel was in 1950s Research Triangle Park, North Carolina. There's this national lab, 2 chemists, Mansukh Wani from India, Gujarat, 1950s में, and Munro from New Zealand. They were both working there. I mean, US land of immigrants. वो lab में जाके वो दोनों काम कर रहे थे. Mansukh Wani was in Munro's lab. His whole job was to extract natural products from around the world, जहाँ से भी उनके हाथ लग सके, और उनको उस समय whatever few cell lines were available, just testing those cell lines. the compounds, extract, not the individual compound. Remember, 1950s, ये technique chromatography ये सब कुछ available भी नहीं थे. So they will test against the cell and see whether it's working or not. Anyway, long story short, they tested many thousands of these extract from various plants from around the world. So my lab, for example, used to be at National Cancer Institute. National Cancer Institute has natural products. वहाँ पे जो chemists हैं, they work with people pretty much around the world. वो एकदम पाताल तक जा रहे हैं, ओशियंस के अंदर जा रहे हैं और वहाँ से नेचुरल प्रोडक्ट निकाल रहे हैं. वो कहाँ कहाँ अमेज़ॉन में जा रहे हैं, वो हिमालय में जा रहे हैं, ढूंढ के वो निकालते हैं, फिर उनको टेस्ट करते हैं. And there is one of the largest collection of natural products वहाँ पे. So these are what natural product chemists they do. So anyway, they looked at and they identified a compound which was from the bark of the yew tree, state of Oregon, Seattle के नीचे जहाँ से अभी मैं हूँ, स्टेट ऑफ ऑरेगन में. तो वहाँ से एक ट्री उनको मिला, उसकी जो बार्क है, उसकी जो स्किन है, बार्क है, उसके एक्सट्रैक्ट से उन्होंने जो किया तो कंपाउंड दैट एक्सट्रैक्ट स्टार्टेड वर्किंग. तो इट शोड वेरी गुड रिजल्ट्स. दिस इज़ फ्रॉम 50 से लेके मैं आपको एक आध मिनट के अंदर पूरे 40 साल का 35 साल का काम बता रहा हूँ. तो दे एक्सट्रैक्टेड, दे कोलैबोरेटेड, दे सेंट देयर कंपाउंड्स टू नेशनल कैंसर इंस्टीट्यूट इन वाशिंगटन डी इन बेथेस्डा मेरीलैंड एरिया. दोज कंपाउंड्स वर टेस्टेड और eventually it went through preclinical studies जो I've been telling you, preclinical studies develop in उस समय 80s में पूरे 70s में 80s में जितना पता था उसके बेसिस पे, क्योंकि आपकी मॉडर्न क्या है, मॉडर्न जो आज है कल ये पुराना हो जाएगा क्योंकि कुछ और चीज़ें आ जाएंगी. So at that time whatever the best of knowledge was available, they used that, they employed that, and there were 50 clinical trials funded by the government were run. Eventually that compound came, and this is the compound, this is the structure of paclitaxel. Now if you look at the paclitaxel structure, there are so many chiral centers. It's not easy to synthesize the pure isoform. I mean, it can be synthesized. At that time there was no synthesis available also because we didn't know the structure kya tha. So then government floated, दो tender निकालते हैं, BMS, Bristol Myers Squibb gave the best tender, they got to start making. Clinical trials were on 1992 के अंदर the compound was approved, which became Taxol or paclitaxel जो है. This was for ovarian cancer for women. Compound gets approved 92 के अंदर और 94 के अंदर the reports come कि it's really not working. It works only in a small percentage of women. What happened? 40 साल लगाए, बिलियंस ऑफ डॉलर्स लगाए, जितना उस समय मॉडर्न था वो टेक्निक्स को, उस नॉलेज को यूज़ किया, बट इट्स नॉट वर्किंग. इट वर्क्स ओनली अ स्मॉल फ्रैक्शन ऑफ पेशेंट. ओके. Subsequently, so obviously the best of the brains, biologists, toxicology, pharmacologists, chemists, they got involved. कि हुआ क्या? Why it's not working? Now let's try to find out. It turned out, so one of the person who was very active and was the toxicologist on there was Susan Horowitz. She was a faculty in New York. Started look at ये है क्या? The drug was developed for targeting beta tubulin. DNA damage करता है, वो DNA को damage करता है. That was a presumably mechanism of the drug. And now these are beta tubulin जो हैं, these are जो आपके cell body में, cell के अंदर, you'll start to appreciate कि nature कितनी मजेदार है, कितनी beautiful है. Cell के अंदर एक protein है ये, जो 23 nanomolar, you can't even see through your eyes. There are 3 types of tubulins, alpha tubulin, beta tubulin and gamma tubulin. Gamma tubulin का function कम है. Alpha, beta, gamma is based on their structure, orientation, कौन से acid, amino acid कहाँ लगे हुए हैं. So and that combined forms what is known as microtubules. Those microtubules are formed. उन microtubules के सैकड़ों function है, cell के अंदर, tissues के अंदर, organ के अंदर, body के अंदर के, इसलिए हम सब अपना अपना काम कर रहे हैं. So that's only one of the proteins. So just imagine how many different types of proteins are in a cell, and each one connecting with each other. It's an ocean. Cell is an ocean of protein. So it was developed for this beta tubulin. So and now there are, I'll come back to this. There are 28 drugs in the market trying to target beta tubulin. Anyway, and I'll come to that. So when she started, she and her team and other those biologists who were involved started to look at the drug was supposedly, so let me again show you this structure. ये beta tubulin alpha and beta जो है, see I have written there alpha and beta tubulin form, they come together and form these tubules, columns बनाते हैं. They beta tubulin जो है उसके अंदर जो कुछ एक प्रोटीन्स हैं वो थोड़े बाहर को एक्सपोज होते हैं. Now each tubulin, alpha tubulin, has many different types of protein just to make alpha tubulin. Each of those protein, so as a chemist to appreciation के रचनाकार की रचना कैसी है, एक बीटा ट्यूबुलिन जो है जो कि स्मॉल एक प्रोटीन है विद इन द सेल, बाकी प्रोटीन्स की बात नहीं कर रहा, that is formed by 3 alpha beta gamma tubulin. Each one of those is a sequence of amino acids. उसके अंदर सीक्वेंस का भी किस ऑर्डर में है, that gives the shape of alpha beta gamma, right? Alpha and beta, they have kinase, GTPase, different type of proteins within that protein to form the bigger protein. And so they come together to form एक तरह की tube, nanotubes, microtubules उसे कहते हैं हम. And alpha and beta वो एक दूसरे से tightly packed होते हैं. उसमें से कुछ एक proteins हैं, naturally that's how it works. But some of this, and I'll come to that, let me not jump ahead of me. So beta tubulin को target करने के लिए presumably this drug was which works on beta tubulin. जो paclitaxel है, presumably taxanes work on यहाँ पे, you see my pointer here, वो यहाँ पे काम करते हैं. That was the understanding when it was developed, which is still correct. But the question is, जो single beta tubulin है, it turned out it has 6 different isoform. वो 6 different तरीके से orient हो सकता है, just single beta tubulin. Depending on which patient, उसमें कौन सा isoform of tubulin is overexpressed, this drug will work or not work. So beta tubulin 1, beta tubulin 2, beta tubulin 3, beta tubulin 4, 4A and 5. There are 6 isoforms. So and then she studied one of these studies to show किस तरह से कौन सा isoform ऊपर या नीचे होता है, किन which patients responds and which patient do not respond. They started to look at उन patient samples को, which ladies responds and which don't. It turned out the ones who did not respond to the drug had beta tubulin 3 and 4A, उनमें वो ज़्यादा overexpressed था protein, and those were about 2/3 of the women, those who were not responding. So we started to see what influences beta tubulin 3. So my lab, from looking from the chemistry angle, we started collaboration with somebody who was a tubulin expert, someone who had MD and PhD, clinician and scientist, his whole career was only beta tubulin. We started to look at कि beta tubulin क्यों नहीं काम करता, और specifically beta tubulin 3 now, not just other beta tubulin 3 isoform. Just to give you an idea and appreciation कि how nature is complex, just beta tubulin, ये उसका जो मैं आपको दिखाने जा रहा हूँ is the simplified version. This is the simplified version of that. Now this single beta tubulin has many GT pathophysiological which play in microtubule dynamics के अंदर उन proteins का role है. There are at least 19 different kinases जो उसमें involved है. ये सब मिला के एक beta tubulin 3 सिर्फ बनाते हैं, बाकी हजारों प्रोटीन की तो बात बाद में होगी. So how complex it is. So it's a challenging task. So we started to look at it, how do we understand? There are no tools available, how do we understand? So we started another journey, try to look at patients का sample लेके, and to realize to understand कि उनका makeup क्या है. No, this one cut. Yeah, don't show this one. Okay, this I'm asking because the data is not published at the patent application file. So we looked at 5 patients. They have gone through at least 4 different type of treatments, and still the cancer came back. So we're curious to see कैसा हुआ क्या, why, what is makeup of their उस biopsy के कारण कि 4 different type की treatment उससे हो चुकी है, but it comes back. So we did, now you see analysis, we did micro RNA analysis looking at the micro RNA level पे कि कौन कौन से signaling pathways, कौन से proteins जो हैं वो over expressed हैं. So you see another technique that we use. I showed you about synthesis. हमने किया, हमने biopsy ली. I'm using the terms you hopefully now you can connect जो पिछले 4 दिन से बता रहा हूँ. We took the patient samples, did the analysis. They have gone through 4 different types of treatment, and this is what happened. So what we observed was that there are pathways which are associated with microRNA processing and stability of the protein. वो जो pathways हैं, those pathways are significantly elevated in those patients. So you simply देखो कि उनके अंदर वो जो pathway हैं, अब there are thousands of those. उनको जब micro analysis करते हैं तो देखिए कौन से pathway, which pathways are before and after treatment. So we identified those pathways. Okay, that's those pathways are there. And so this is again 5 patient tumor samples associated with this. And then we looked at various different pathways. One of the protein that stood out as a pattern, जो I've been saying AI and pattern, machine learning and pattern, this is real data. और उसमें देखा कि one of the protein, processing protein जो उन pathways के अंदर काम करता है, इसे Y-box protein 1. It's a cold shock protein. It functions DNA associated also with RNA. So it's a sort of a transcription factor. So my terms which I'm using, if you do not follow, try to read them. It's a transcription factor. जो transcription होता है ना, जो आपको फिर से S phase, G1 phase, M phase, G2, वो transcript जो है आपका DNA की जो information है, RNA उसमें जाके transfer करते हैं नए cell के बनाने में, जो transcription part है. So there are many proteins that play. One of the protein is Y-box protein 1. So it interacts with DNA, it interacts with RNA. That's what its roles is, and many functional जो हमारे cell के अंदर, tissue के अंदर आपके body की functions है, it plays an important role. Now it is, for example, a few that I mentioned, it's a key regulator of RNA processing. Multifunctional cold shock protein regulates DNA and RNA in the cellular events. Cell के जो events हैं, I have listed here, for example, and now once they splice transcription होता है उसकी packaging में किस तरह से वो package होते हैं, नया cell बनता है, उन सब के अंदर Y-box protein plays an important role. And here is, so this is my PhD student who put together this whole, looked at the entire literature to see Y-box का role कहाँ है और क्या उसके नीचे ऊपर दाएं बाएं, what are the different pathways which are associated with it and how it gets influenced. So these are just simplified version. So it regulates within the cell many functions, and here the EMT जो है metastasis जो सेल से बाहर निकल के फैलता है, मेटास्टाइज होता है, उसको फंक्शन को, apoptosis killing the cell, right? ड्रग efflux पंप जो मैंने बताया उसमें भी उसका रोल है. इट जो स्ट्रेस होता है, जब बॉडी के ऊपर कोई स्ट्रेस, सेल पे जो स्ट्रेस होता है उसके रिस्पांस में इम्यून सिस्टम के अंदर इट हेल्प्स टू फंक्शन. सो अगेन सेल साइकिल के कंट्रोल करने में उसका फंक्शन है. सो दीज आर some of the factor and signaling pathways. So one single protein, inside the cell, it has so many different functions. This one protein, just imagine that protein is connected to another few protein, large protein, I don't know how many small protein. Each one has its roles to play within the cell, within the tissue, within the organ. वो सब जब बन के तैयार होता है, ये सुंदर रचनाएं पैदा होती हैं. It's a very complex system, right? So we started to look at के अगर ऐसा है, and there are various, these whatever, EGFR, IGFR, ये जो different pathways या signaling हमें पता है, like ये as best as we know, tumor microenvironment factors क्या क्या हैं, उसका drug efflux क्या होता है, EMT, इसके जो known proteins हैं, those who contribute, those are, as you see here, directly or indirectly are affected. So they are upstream to this, some of they are downstream to this protein. So we thought, okay, there is probably an opportunity. Okay, we start understanding this protein and try to design molecules which may interfere in the function of this protein in the cancer cells, so that cancer cell का जो है growth को कर दे. Now, before we started, we said, okay, let's first look in the literature and see what do we know, what is reported in the literature. So what I'm showing here is we looked at two databases, and what it shows is this is collection of patient, ovarian cancer patient, उनका YBOX1 protein expression. And 1, 2, 3, 4 here are the stages. And stage 1 cancer, stage 2 cancer, इस तरह जो stages हैं, उन हर stage पे YBOX protein उनका expression क्या है. So as you see here, as it advances, YBOX protein becomes more and more active, even though after the treatment, which means it acquires resistance. So this is an acquired resistance. The more treatment होती जा रही है, वो और भी aggressive होता जा रहा है. That is one form. So and we looked at another database. ये the cancer science सिंगापुर का है. Again, as you see, as the growth, the stages उसकी बढ़ती हैं, उसका expression बढ़ता जाता है even after treatment. So which means treatment से कोई फर्क नहीं पड़ता. Protein is something. We try to look at now. Okay, what are the proteins? Now ये तो ठीक है, YBOX protein is a complex. What are the proteins which go up and down when you after the treatment? So we identify few of these. CD44 is one molecule. MYC, c-MYC is a master regulator, one of the master regulator. There are many master regulators. P53, c-MYC, YBOX, snail, PIM1. They are kinases. We try to identify कौन से ऐसे हैं जो कुछ समझ में आए, so that you start preparing and start working around that. So and then we look at again okay, if YBOX1 is so important, so maybe there is an opportunity that you start developing a drug for targeting YBOX1, because अगर उसको आपने control कर दिया तो बाकी की जो उसकी जो functions हैं which influences the cancer growth, उसको रोक देंगे. So before that we again looked at the patient data to see कि क्या हो रहा है, and it clearly showed कि patients where there is a low expression of YBOX, they are responding better to the treatment compared to where there were a higher expression of YBOX1, which correlated with our hypothesis theory that targeting YBOX1 could be a way to develop a drug for this. Now, go back and think to my first and second and third lecture. Okay, you have to first identify the target आपका क्या है. So we looked at the cells. We went through all the data that we could from the public domain, from the patient data or research data, and tried to make sense of. We did the analysis of the patient samples, try to understand at the microRNA level what is overexpressed in those patients, go back to patient sample data, उसको correlate करने के लिए कि really it is making sense या हमारा भ्रम है या there is something else to this. So when that correlation, all of that happened, now that gives you confidence कि ठीक है, we are on probably ठीक track पे हैं करने के लिए, and that's how piece by piece we put the information together. So now this is to correlate कि वहाँ तक पहुँचने के बाद में okay, from the patient data it shows कि जिनका high expression है, they respond poorly, which means this is a potential target जिसके लिए आप drug develop कर सकते हो. Simultaneously, my lab has been interested from the chemistry point of view. As a chemist, we've been interested to see drugs when they went to clinical trial, phase 1, phase 2, and they failed. They didn't go to market. तो उसमें क्या change था? What was wrong with their structure? Or so we were trying to, again, as I've been telling you guys, from the lens of chemistry, इसके structure में ऐसी क्या issue है, क्या problem है, that we can fix, so it becomes effective. So we started to look at those drugs. One of those drugs was etoposide. Etoposide is a natural product. It is a compound. These are podophylotoxins. If you look at podophylotoxins are class, so this is a drug which is obtained from there are plants वो ठंडी जगह पे होते हैं. यहाँ पे फुटिल ऑफ हिमालयस नॉर्थ ईस्ट इंडिया में होते हैं. इट्स अबाउट अ मीटर हाई प्लांट जो है, एंड उसके जो उस प्लांट में से पोडोफाइलोटॉक्सिन्स होते हैं. और या फिर नॉर्थ ईस्ट ऑफ यूएस कनाडा जहाँ पे बहुत ठंडा वेदर होता है, वहाँ पे लगभग 1 मीटर जितने और 3-3.5 फीट के होता है ये प्लांट. उसके जो है डिफरेंट पार्ट्स में से ये कंपाउंड्स आते हैं. तो उसमें से इटोपोसाइड निकला था 80s के अंदर. As I told you, the natural product chemists are extracting and then they are testing. Etoposide turned out to be one of the compound which was effective. But for whatever reason, initially it was withdrawn. So, but showed toxicity, and again it came back with uske analogs bane and it started to come back. So we were interested कि ठीक है उसकी toxicity remove करने के लिए as a chemist what is it that we can do. So this is basically a general structure which has 4 rings, and then ये X R1 R2 all these substitution. We as a chemist started to design our own variation to see कि उसकी toxicity जो है उसको. Now toxicity उसका the reason was this right side. So when it breaks metabolite, okay, when etoposide goes inside it metabolites, enzyme उसको break करते हैं. One of the product you obtained is catechol. आपने catechol का नाम सुना है? It's a simple chemical. It's acidic. So सीधा जब जाता है तो साइड इफेक्ट उसका होता था अल्सराइटिस, अल्सर होने का, क्योंकि इसे हाईली एसिडिक कंपाउंड. सो वी थॉट ओके ठीक है, अब इसके जो कैटेकॉल की प्रॉपर्टीज़ जो होती है उस मॉलिक्यूल के रिंग का स्ट्रक्चर थोड़ा चेंज करते हैं. सो वो अंदर जाके बनेगा, वो कैटेकॉल नहीं बनेगा, इट वॉन्ट बी एसिडिक. सो वी दिस इज़ हाउ स्लोली स्टेप बाय स्टेप वी स्टार्टेड टू लुक एट कि किस तरह से हम उसको चेंज कर सकते हैं, किस तरह से हम उन चीज़ों को चेंज कर सकते हैं. वी स्टार्टेड टू डू दैट and try to come up with our own structure, newer structures. We did that and then made compounds, and we tested on 60 different cancer cell lines, 9 different tumor types: head and neck, renal, breast cancer, leukemia, ovarian, prostate cancer, colon cancer. So 9 different types we tested, and these are the the cell lines which are mentioned here. And it turned out this was separately happening. We didn't go for this project. We were separately as a chemist interested to see why drugs were toxic and not working, and we designed that. And we designed compounds which turned out to be highly effective against the ovarian cell lines. So we said, okay, from that project, from the chemistry studies, we identified, we designed a compound which is effective. Here we have shown in the ovarian cancer models that there is a protein that doesn't work. What if we start using this against these cell lines, ovarian cell lines, these compounds which we have developed, we identified, and we started testing the research part. So we are researching. Long story short, we identified the compound which were highly effective, and then we started to do in vivo studies. Took into the mice, implanted cancer cell lines, ovarian cancer cell lines, did that, and you see this, the colors that you see, we did imaging to see where the tumor was. So how many things I have told you so far, जो मैं पिछले 4 दिन में कहता आ रहा हूँ. Chemistry, microRNA analysis, assay development, right? Imaging techniques that we used, right? So we did all of these things, and these are the cell lines that we developed models against which we started testing these compounds. And you see, ये जो है control, which is without control, which was used as a reference, असर कुछ भी नहीं होता. When we give our drugs, you see the tumor is suppressed. It is suppressed further. So we did these studies, mice 42 days of the treatment. Now, these words here, 10% PEG 300 insulin formulation. We prepared the formulation सिर्फ DMSO में नहीं. formulation that is not toxic to the cell, formulation which is tolerable by the cell, but formulation in which your compound is soluble. Formulation chemist. That and making it bioavailable. Because if formulation, whether it will take it to the site of action. If it doesn't, it's not going to be helpful. So you see how many of different expertise जो मैंने आपको बताया पिछले 4 दिन में मैं कहता आ रहा हूँ. And then this is before treatment with the vehicle, after treatment, tumor shrink. With another compound and we use 20 mg per kg. Right? And you see with and the second compound even further it's suppressed. So then we did the pharmacokinetic studies कि कंपाउंड देते हैं तो अपने ऑर्गन साइट तक पहुंचने में कितना टाइम लगता है, उसका हाफ लाइफ क्या है. You see, I'm using the term which I've been telling you for last 4 days कि ड्रग का क्लियरेंस का हाफ लाइफ क्या, because it has to reach to the site, and we did that. You see here liver जो है these are again PK analysis, जो C max जो है 1 point, जो maximum concentration after you give the treatment कितनी होती है उस ड्रग की. So you can do again chromatography, do the analysis which tells you can get up to 1.6 microgram per ml. Then you have to study and see is this enough to get the effect that you need. So you all of these Tmax, how much time, so highest concentration you get, what time it takes to get to that concentration. 5 मिनट के अंदर ही उसका असर आया था. So drug is effective. You give it and 5 minutes के अंदर उसका max concentration प्लाज्मा में निकल आती है. T half कितनी देर तक उसमें रहती है, right? All the things that I've been telling you. So we studied that to find out कि कितनी रहता है. And then drug distribution, कि drug reaches to कौन-कौन सी sites में. So then these are the standard. You look at the organs, especially liver, ovaries में, क्योंकि because it was for ovarian cancer. Liver में क्योंकि liver के अंदर liver metabolites उसको chop करते हैं, right? So you find out that concentration, and then toxicity. AST, ALT, these are toxicity for liver, और जो है आपका कि कितना वो toxic है. And basically what you do is you calculate the enzymes. वो enzymes से you can quantify कि उसका enzyme level क्या है, उसकी toxicity क्या होगी. So we did all of this liver toxicity for again 42 days. These and look for the enzyme. And this is after many years of work. We published this in this 2021. When we published, soon after that, around that time, a group in Harvard was studying this. They saw immediately in उनका जो reference, जब उनका पेपर रिव्यू के लिए आया, उन्होंने फिर से हमारा पेपर लेके उसमें डाला कि अब तक सिर्फ यही ड्रग है जो काम करता है. There is nothing available, because they were also independently studying Ybox1, and this is the only drug which is reported that works. So this is just to give you an appreciation of from chemistry to assay development to model development, and you see how many terminology that I used. All of these are done to identify the candidate and then do all the PK studies. This is again ADME PK studies जो हम कर रहे हैं. And then we started to look at, okay, now this candidate, let's go further and try to see what it does. So we expanded that to see it works in ovarian cancer. Ybox protein 1 is expressed, and I showed you those chart, patient chart, as with the stages overexpression बढ़ जाता है. But then see कि Ybox protein 1 है और दूसरे कौन से कैंसर टाइप हैं जिसमें वो प्रोटीन का फंक्शन है. हो सकता है ये ड्रग उसके लिए भी काम करे, whether it's a breast cancer or head and neck cancer, lung cancer, जो भी है. So we started to look at all consequences. तो हमारे in my lab we're doing a lot of work on triple negative breast cancer. So we had models available. We said, okay, let's now look at triple negative breast cancer. We looked at that and we identified that there were actually even TNBC, triple negative breast cancer, also Ybox protein is highly expressed. So you say, okay, we have molecule, we have models, now let's start testing that. So this is the in vitro study that we did, various specifically breast cancer cell lines बनाई, models बनाए, उनके अंदर test किया. When it worked out, we went ahead and then created the various stains, blacks, 6 mice, लेके उनके ऊपर we did various these studies again to show 93, 92 जो two compounds है किस तरह से काम करते हैं. And as you see यहाँ पे you probably may not be able to see ये metastasis you can control. So sorry आपको नज़र नहीं आ रहे होंगे because of there are these arrows which actually show कि उसका effect क्या है in control condition पे. So metastasis is when it moves from one place to another place, and you can follow the enzymes. Ki67 is one of the enzymes. You follow that and it can tell you क्या Ki67 बढ़ रहा है या कम हो रहा है. You can see before and after treatment. You can tell whether your compound is working or not. So we took this, we tested in the lung metastasis studies के अंदर. So this compound was effective. This was the picture may not show, but look at the quantification. Vehicle के अंदर really doesn't work. बहुत metastasis होती है कि ड्रग जो कैंसर सेल हैं वो escape करके दूसरी यहाँ पहुँच जाते हैं और बढ़ाते हैं. When we gave our one candidate, it suppressed metastasis. When we gave the another candidate, it suppressed further, which means the drug on attacking Y-box protein 1 was keeping it controlled locally rather than spreading. So you do these kind of study, and then we did a lot of studies to establish that the drug target is actually Y-box protein 1. So there are many different assays that you could use. So this is an assay which is called SETSA. जो है the circular the thermal analysis, which is basically what is done is you take your protein of interest, and then you take the compound, you take a group of proteins and put the compound, and then you turn the stability of the protein. And if the protein, whichever protein जिसके साथ में आपका ड्रग इंटरैक्ट करता है वो प्रोटीन को पकड़ के रखेगा, तो उसका जो थर्मल शिफ्ट है वो रुक जाएगा, कम होगा. So you look for the proteins and then say okay, my drug or this drug इस प्रोटीन के ऊपर सबसे ज़्यादा शो करता है, which means that's the interaction mode. So you identify which proteins are affected by your drug. So that's called SETSA analysis. We did that, and we established that Y-box protein 1 is actually the target. We did many, again, proteomic studies, genomic, whole cell genomics, proteomic studies to identify कि even with 2 mg per kg, how with the time dose dependence, कितने टाइम में उसका असर होता है, कितने डोज पे उसका असर होता है. So then you actually now is the development part. Once you have the discovery part, now is the development part to see how the drug can be developed. And this is all you see the proteomics to establish that the drug is working against that protein that you intended it to work on. Now, as I showed you, our interest was कि paclitaxel. My story started from why paclitaxel के अंदर resistance है. So research तो कर दिया. We started with the hypothesis कि Y-box protein जो है is the culprit जो टैक्सॉल की जो है एक्टिविटी को लो करता है. अब अगर हमने नया ड्रग बनाया है तो भी उसको प्रूव करना पड़ेगा ना कि वाकई उसको रोकता है. Otherwise just a theoretical part. So what we did was, as you see here, we studied, these are 2 different, OCAR8 and SCO3. They are 2 commonly used ovarian cancer models, ovarian cancer cells and 2 different types के. उनके अंदर टेस्ट किया, identified कि डोज ये paclitaxel का डोज है, and this is only combination study. So you look at this, when the drug which is in the market, जो कि टैक्सॉल जो यूज़ होता है, when you give this drug, टाइम के साथ में जो है वो उस पर ज़्यादा असर नहीं करता है, वो बढ़ता रहता है. रेजिस्टेंस के कारण ट्रीटमेंट का असर नहीं होता, जो है द कैंसर जो वॉल्यूम है उसका बढ़ता जा रहा है. व्हेन यू गिव द कॉम्बिनेशन विद द न्यू ड्रग, इट सप्रेसेस, उसको नीचे ले आता है. दैट गिव्स अ प्रूफ कि ठीक है तुम्हारा आपका जो है ड्रग काम कर रहा है. नाउ दिस इज अगेन इन xenograft model. Remember, mice, and then I will come to patient-derived xenograft model. You take the patient sample, implant inside the mice, let it grow. So it's a human patient sample which is inside the mice. And then you treat that to see now it is working in the patient tissue cells or not. So we did that. And then another thing is to toxicity and to metastasis. Now this is one control जो ट्यूमरॉइड्स स्पेरॉइड्स जो बनते हैं वो उसका वॉल्यूम कितना है, कितने बनते हैं, दैट्स अनदर वे ऑफ इंडिकेशन ऑफ कैलकुलेटिंग वेदर योर ड्रग इज वर्किंग ऑन आर्ट्स. सो एज यू सी हियर कंट्रोल जिसमें कुछ भी नहीं दिया गया, पैक्लिटेक्सल जिसमें दिया गया फिर भी उसमें थोड़े भी बच जाते हैं, ड्रग अलोन उसमें थोड़े भी काफ़ी वो साफ कर देता है, थोड़े बच जाते हैं. बट व्हेन यू गिव द कॉम्बिनेशन ऑफ दिस पैक्लिटेक्सल एंड EC56 एज यू सी हियर significantly changed और यहाँ से लेकर उसको सप्रेस करके यहाँ पहुँचा दिया, right? And if you look at this combination, control, our drug alone, paclitaxel alone, but when you give the combination, वो उसको एकदम सप्रेस करता है, जो ट्यूमरॉइड्स जो हैं, ट्यूमर साइज एकदम से सप्रेस करते हैं, which means it shows that in combination of the new drug, your paclitaxel, which is the drug used in the clinic, combination में lot more effective होता है. And we have shown that it happens because the drug, so in a very simplified way, what I've been telling you is कि जो Taxol जो है उसका resistance होता है because of Ybox protein 1, YBX1 protein. If you take a drug जो culprit है, जो गुनहगार प्रोटीन है, जो प्रोटीन which is the driver, आप उसको suppress कर देंगे तो जो आप commercial drug ले रहे हैं उसकी एक्टिविटी बढ़ जाएगी, फिर से वो काम करना शुरू कर देगा, correct? And that's what you see here. Okay, because when you combine, why combination is working? Because when you give the new drug, the drug that my lab has been working on, if you give that drug, उसने Ybox protein 1 उसको दबा दिया, अब अगर आप पैक्लिटेक्सल देते वो काम करता है. तो जो ट्यूमर वॉल्यूम है वो ज़्यादा से लेके एकदम नियरली यहाँ से देखिए, this is without treatment, with combination treatment, it is suppressed. So that gives you as a scientist more confidence कि आप ठीक दिशा में जा रहे हैं. You are, and then we expanded those combination studies again, as you see here, as you see again imaging. We use the imaging to follow, to study how much it's affecting. So you see control, single drug, when you do the combination, as you see the volume goes down. Okay, so we tested the same thing. There are various different types जिसके अंदर जो है ये Ybox protein का expression. This is TCGA data, the cancer genome atlas. It's called TCGA, cancer genome atlas. That data shows, which is, it keeps on the NCI or NIH जो है, they collect information from patient information, they keep on updating and they keep on analyzing mRNA level के ऊपर. So then we looked at which are the other ways, and then we identified again TNBC के अंदर है, which I started to tell you. We looked at TNBC, same thing, we studied in vitro study, again to look for what are the drivers, which proteins go up and down before and after treatment. Again, Ybox protein turned out to be one of the driver. And then, as I was telling you, showing the structure of beta tubulin, how many GTPases, how many kinases, it's a complex soup. To identify कि Ybox protein actually कहाँ पे काम करता है, there are many mRNAs, there are many mRNAs there, right? So we do that, and then one of my postdoc spent nearly a year and a half एक RNA को पढ़ने के लिए, एक mRNA को पढ़ने के लिए. And then fortunately for us, when we were working with this, so one of the Nobel laureate actually had in his Nobel lecture mentioned about this protein. And then we worked on that, and we finalized, and we sent him compound, and he's looking at the structure, at the mechanism of that. So anyway, we narrow it down to one RNA, because RNA is small RNA, large RNA. कौन से RNA पे actually Ybox protein का असर है. We did a lot of these studies to narrow it down to what could be the mechanism. So I guess in the interest of time, these are the people who worked. I'm just standing here and talking. And with that, so I hope you all get the point. And people were asking me yesterday about contact में क्या प्रोफेसर गुरजसप्रीत सिंह से तो मिल ही सकते हैं, मैं उन्हें तकलीफ ना दें इसलिए मैंने यहाँ इसमें दिल लगा दिया है. Feel free to reach out to, if you have any question. Okay, so I hope in past few days, as I was trying to tell you, and now this is experimental. Therapeutics is experimental. I hope as I was able to show you how this whole enterprise works. It's a multidisciplinary effort. A lot of expertise, lot of resources, lot of मशक्क़त करनी पड़ती है बहुत intelligently, but it's a lot of fun. आप कोई नई चीज़ का आविष्कार करते हैं, नई चीज़ बताते हैं, which helps even to move the science forward. It's a lot of fun. It gives you satisfaction, it gives you more motivation to continue. And today, for example, I was trying to show you कि various different techniques, modalities, disciplines, they come together एक मर्ज की एक दवा निकालने के लिए, and you hope it works and it helps. So with that, I'll pause here. Wish you all very best. Feel free to reach out to me anytime if you need any help, if I can be of any help. All right. Thank you. And निकाल दो.
Thank you so much, sir, for that insightful and comprehensive session. We now have a short tea break.
आपके सामने कॉपी किया था मैंने. फिर करता हूँ. मैं भी करता हूँ. I'll just do it right now. Yes. Thank you so much. आज आपकी चाय जरूर पियेंगे.
Welcome back, everyone. Now we have Mrs. Priyanka, a research scholar under the supervision of Professor Gurjaspreet Singh, to present a lecture on the therapeutic role of triazoles in ulcer management.
Thank you so much, Tanya. A very warm good morning to everybody present here. I am Priyanka, Research Scholar of Department of Chemistry, Panjab University. Today, I am going to present a topic that is the therapeutic role of triazoles in ulcer management. As we all know, the stomach is very important and crucial organ of our body. That's important role is in digestion of food, and the stomach's main function is to store food and partially digest it through a combination of physical churning, digestive juices and muscular contractions. We have very common stomach diseases like gastroenteritis, that is the infection and inflammation of the stomach and intestines. Next we have hiatal hernia, that is the part of the stomach pushes up through the diaphragm. Gastroenteritis, that is the delayed stomach emptying. Gastritis, that is the inflammation of the stomach lining. Next we have peptic ulcer diseases. We will discuss the peptic ulcer disease in detail in the next few slides. Basically, ulcer is the open sores in the mouth, back of the head, shoulder blades, heels, hips, elbows, ankles, back of the knees, toes, feet and ears. These I have discussed are the pressure ulcers. That are the reason for these ulcers is the bedridden conditions of the patients for a long duration of time. Next, we also have ulcers in the lining of the stomach or the duodenum. That is the first part of the small intestine. Peptic ulcer disease is the broad term that is used for the stomach ulcers and duodenal ulcers. Stomach ulcers is the name that is used for the ulcers that are located in the lining of the stomach, and duodenal ulcers are found in the lining of the duodenum, the first part of the small intestine. So we will discuss the stomach ulcers in detail. Stomach ulcers are the ulcers that are present in the stomach lining. It's a common cause of central stomach pain, pain coming from a very particular spot, often with a burning. But not all stomach ulcers cause noticeable symptoms. Stomach ulcers are very common and treatable, but they can be life-threatening if they are not treated timely. Some can continue to erode through the stomach wall until there is a hole, and some can bleed continuously over the time. We can have very common symptoms for the stomach ulcers like indigestion, vomiting, blood, nausea or recurrent upset stomach, loss of appetite, abdominal bloating, burning, etc. Among the causes of stomach ulcers, there are some very common, the major stomach ulcer causes are H. pylori infection, and the next one is NSAIDs. We will discuss these two in detail. First is H. pylori infection. H. pylori is a very common bacterium infection that is found among half of the people worldwide. Sometimes it's overdose in the stomach. As the bacteria continues to multiply, they eat into the stomach lining, causing chronic inflammation that leads to gastric ulcers. Among the causes of H. pylori infections, we have very common causes like person to person. H. pylori can be spread through oral contact. It can also be caused due to the contamination in food and water, that is the unhygienic conditions of food and water. Next is living conditions. As we all know that the conditions in the developing countries is very overcrowded. So the overcrowded living situations and limited access to clean water can also lead to H. pylori infections. Next, hygiene, family history and lifestyle factors like smoking and prolonged use of NSAIDs, consumption of acidic food regularly like by the upcoming or these young generations of the life. Next we have NSAIDs. NSAIDs are the non-steroidal anti-inflammatory drugs like ibuprofen, naproxen and aspirin. The prolonged use of these medications to come back from the pain can irritate the stomach lining on contact, and they also inhibit some of the chemicals that defend and repair stomach lining like bicarbonates. We also have some very less common causes of stomach ulcers like smoking, alcohol, consumption of spicy and acidic foods. Then we have diagnosis and test. Once we are aware of the stomach ulcers present in our stomach, the very basic treatment of this ulcer is the endoscopy. In endoscopy, an upper endoscope goes inside the stomach with a very tiny camera on a long thin tube. This tube goes down our throat while we are under sedation. As we all know, that food is essential for sustaining life, providing the energy, building blocks necessary for growth, repair, and overall bodily functions. Nutrients in food, including carbohydrates, proteins, fats, vitamins, and minerals, play a crucial role in maintaining health, preventing disease, and supporting this immune system. So we have some foods that should be emphasized to prevent ulcers, like the foods that are rich in fiber, probiotics, lean proteins, and those foods which are low in acidity are generally well tolerated and can aid in healing and symptom management. On the other hand, we also have some foods that should be avoided once we know that we have ulcers in our stomach. These foods are spicy food, acidic foods, fatty and fried foods like citrus fruits and tomatoes, caffeine, alcohol should be avoided as they irritate the stomach lining, they increase the acid production and delay the healing process, which worsens the ulcer symptoms. Once we know that we have ulcers, we can use some medications according to the different type of ulcers present. If the reason of ulcers is the H. pylori infection, we can have medications like tetracycline, metronidazole, clarithromycin, amoxicillin. Next, if we have ulcers due to the poor protection of the stomach lining, then they can have medications like sucralfate, misoprostol, bismuth subsalicylate. Next, we have histamine receptor blockers like famotidine, cimetidine, nizatidine. Next, we have proton pump inhibitors, like the medications which will inhibit the use of proton pumps in the stomach lining. These includes esomeprazole, dexlansoprazole, lansoprazole, omeprazole, etc. We will discuss some of the medications one by one in detail. First is tetracycline. Tetracycline is a bacteriostatic antibiotic that reversibly binds to the 30S ribosomal units. Specific functional groups on tetracycline like dimethyl amino groups, as you can see, amide group and phenolic hydroxyl groups play a role in interaction. These groups are crucial for tetracycline's ability to inhibit protein synthesis by interfering with the binding of amino acyl tRNA to the ribosome. Amino acyl tRNA is the molecule that carries the next amino acid to the acceptor site of the ribosome. Now, due to interference of tetracycline, amino acyl transfer RNA cannot bind to the A site and hence the ribosomal can't move along the mRNA to continue adding amino acids to the growing polypeptide chain, effectively halting the protein synthesis. Next, we have clarithromycin. Clarithromycin specifically binds to the 23S RNA within the 50S ribosomal subunit of the bacteria cell, which participates in the protein synthesis. By inhibiting H. pylori protein synthesis, clarithromycin helps to eradicate the bacteria and reduces the risk of ulcers or their recurrence also. Next, we have amoxicillin. By any medication, by any way, if we can prevent the formation of ulcers, we can use that medication as the ulcer prohibition. Like amoxicillin's beta-lactam ring opens to acylate the transpeptidase C-terminal domain of PBP1A. So it inhibits the synthesis of cell wall of bacteria. Next, we have sucralfates. Sucralfates negatively charged molecules interact with the positively charged proteins in ulcer, forming a viscous gel-like barrier that helps to reduce the acidity of ulcer environment. Sucralfate also absorbs bile salts, which can irritate the ulcer site and hinder healing. Next, we have misoprostol. Misoprostol stimulates the secretion of mucus. Bicarbonate thickens the mucosal barrier and improves blood flow to the stomach lining. The combined effects of reduced acid production, protection of lining of the stomach contributes to the stomach's ability to regenerate new cells and heal ulcer. As we have discussed this category of medications which are used for the formation of mucus lining, and also we have discussed the medications that are very well known for the effect on the H. pylori infection. Next category of medication contains H2 receptor antagonists. Activation of histamine receptors on parietal cells stimulates the activity of proton pump. The proton pump exchanges H positive ions with the K positive ions into the stomach lining, hence creating the low pH environment in the stomach, which can create ulcers more severely. So H2 receptor antagonists block activation of histamine receptors and therefore reduce acid secretion by parietal cells of the stomach. In the same category of medications, we have famotidine. Famotidine, an H2 antagonist, it blocks the action of histamine on the parietal cells, ultimately reducing acid secretion into the stomach. Next we have cimetidine. It also binds to the H2 receptor located on the basolateral membrane of the gastric parietal cells, and hence it blocks the histamine effects. Next we have nizatidine, which acts as a competitive antagonist, meaning it competes with histamine for binding to the H2 receptors. By blocking the histamine signal, nizatidine reduces both basal and stimulated gastric acid productions. Fourth category of medications contains PPIs, that is proton pump inhibitors, the medications which will inhibit the production of H positive ions into our stomach. The gastric acid secretion occurs in the parietal cells via the H positive, K positive ATPase of the proton pump. So this enzyme is responsible for the production of H positive ions into our stomach. So any medication which will prevent its work or its function in the stomach can be used as a medication. In this category we have isomeprazole. As we know, this enzyme is responsible for the pumping hydrogen ions into the stomach lumen, which is essential for acid production. So by inhibiting the proton pump, isomeprazole reduces the amount of acid produced in the stomach. Next we have dexlansoprazole. It also inhibits the action of H positive, K positive enzyme, which can be beneficial in treating conditions like ulcers. Next we have pantoprazole in the same category. It also leads to the decrease in gastric acid secretion, which is crucial in the treatment of ulcers by reducing the corrosive effects of acids on the stomach lining. I have also mentioned some more proton pump inhibitors with their structures like omeprazole, lansoprazoles. As you can see that all these structures have heteroatoms like nitrogen, oxygen, sulfur present on them, which have a lone pair of electrons basically. So these lone pair of electrons bind with H positive ions produced in our stomach. Hence they reduce the H positive ion concentrations in the stomach, increases the pH, and helps in the prevention of ulcers or acidic secretions. Now I'll be discussing triazoles, which are the new emerging medications that can be used for the anti-ulcer treatments. Until now, peptic ulcer, often caused by excessive gastric acid or infection like H. pylori, requires effective therapeutic strategies to promote healing and reduce acid secretion, while traditional treatments like antacid, H2 blockers and proton pump inhibitors, recent studies highlight the potential of triazole-based compounds. So triazole-based compounds are basically the heterocyclic compounds having 2 carbon and 3 nitrogen atoms on the adjacent positions in 1, 2, 3 triazoles, and at 1, 2 and 4 positions in 1 to 4 triazoles. These triazoles can be used as antioxidant, bacterial, anti-inflammatory, antifungal, anti-cancer, anti-tubercular activities can be shown by these triazoles. In recent years, the Nobel Prize of 2022 was awarded to Caroline R. Bertozzi, Morten Meldal, and K. Barry Sharpless for their work on click chemistry and bioorthogonal chemistry. That is the chemistry that can make a bridge between the biological and chemical applications in human body, which includes the development of efficient triazole synthesis methods. We will discuss some different methods of triazole synthesis in brief. First is Hissens 1,3 dipolar cycloaddition. This reaction involves the thermal cycloaddition of an azide to an alkyne. It will give us the two products, that is 1,4 regioisomer and 1,5 regioisomer in equal concentrations. Next is copper catalyzed azide alkyl cycloaddition. This method has an advantage as compared to Hissens 1,3 dipolar cycloadditions, as we can see that the Hissens 1,3 dipolar cycloaddition gave us the two products, that is 1,4 regioisomer and 1,5 regioisomer. This specific reaction, copper catalyzed azide alkyne cycloaddition, gives us only one product, that is 1,4 isomer only. So this is a basic comparison between these two cycloaddition reactions. We can also have different, other than these copper catalyzed reactions, other methods that can be used for the formation of triazoles can have strain promoted azide alkyl cycloaddition. As you can see that in reactants we have sp carbons that are forced to be present into the ring, so they are filling a strain in this molecule. So any activity, any reaction that can decrease this strain can be used for the effective formation of the products. In reactant we have sp hybridized carbons, and in products these sp hybridized carbons gets converted into sp2 hybridized carbons, releasing its strain and gives us the triazoles. We also can use other metals for the formation of triazoles like ruthenium. In 2005, Fokin and co-workers devised an efficient approach for the construction of 1,5-disubstituted 1,2,3 triazoles by ruthenium cyclopentadienyl complex. Next, we used silver catalyst for the regioselective synthesis of 1,4-disubstituted 1,2,3 triazoles at room temperature only. Iridium can also be used as the catalyst for the electron-rich internal alkynes for the formation of triazoles. Next, bicyclopentadienyl nickel catalyst can also be used for the formation of di-substituted 1,2,3 triazoles under mild conditions. Zinc catalyzed azide alkyl cycloaddition. It was developed by Postnikovs and his research group, affording 1,4-disubstituted 1,2,3 triazoles. Now we will be discussing some commercial drugs of triazoles, so you guys can have some idea about the basic use of the triazoles in different type of diseases. First is fluconazole. It is used for the treatment of fungal infections. Next also we have posaconazole. It prevents fungal infections in immunocompromised patients. Basically, who are immunocompromised patients? The patients who are fighting from a very serious disease like cancer. The immune system of such patients get reduced, and fungal infections to such patients is more prone. Next we have turconazole. It is used for the vaginal yeast infections. Next we have letrozole. It is used for the treatment of hormone sensitive breast cancer. Then we have trazodone. It is used as an antidepressant and for insomnia. Then we have rufinamide. It can be used for the treatment of epilepsy. In epilepsy we have strains, sensations and emotions, loss of consciousness and awareness, staring, confusion, sudden falls, etc. So rufinamide is used for this kind of epilepsy. Then we have some activities of triazoles like anti-ulcer. Some triazole derivatives have been specifically investigated for their anti-ulcer properties. For example, a 1,2,3 triazole derivative has been shown to promote ulcer healing in rats by multiple mechanisms, including inhibiting H. pylori, reducing oxidative stress, suppressing inflammation, and blocking acid production. This gives us an idea that certain triazoles can directly contribute to the treatment and prevention of ulcers in the human body. Next is anti-H. pylori bacteria. Some triazole derivatives also show activity against H. pylori, which is crucial for treating and preventing peptic ulcers. They can interact with enzymes vital for the survival of H. pylori in this acidic stomach environment, such as carbonic anhydrase. Anti-inflammatory properties, antioxidative effects. Oxidative stress contributes to the gastric mucosal damage and ulcer formation. As we know that the mucosa is the lining that prevents the stomach lining from getting ulcers or from getting corrosion. So triazoles can also be used as the antioxidant effects, which can help in protection of the gastric lining from such damage. Next we have acid secretion. Some triazole compounds have been shown to inhibit gastric acid secretion and exert cytoprotective effects on the gastric mucosa, similar to the proton pump inhibitors used in the ulcer treatment. They can also be used for the reduction of NSAIDs. NSAIDs were non-steroidal anti-inflammatory drugs which caused ulcers, but often cause gastric ulcers as a major side effect due to the inhibition of prostaglandin synthesis, which are cytoprotective in nature. In conclusion, we can say that the versatility of the triazole scaffolds allows for the synthesis of diverse derivatives with enhanced biological activities, including improved efficacy and reduced side effects compared to some conventional anti-ulcer medications like nizatidine, etc. This broad spectrum of action, coupled with promising in silico, in vitro, and in vivo results, strongly supports the conclusion that triazoles are indeed very efficient anti-ulcer agents with considerable therapeutic potentials. Thank you. Um, Actually, sir, both have approximately stability between 1 to 4. बंद ही हो गया वो उतने में बंद ही हो गया दोबारा फोटो खिंचा लेना
Thank you so much, ma'am, for the insightful lesson. Now I would like to call Ms. Parul, a research scholar under the supervision of Professor Gurjaspreet Singh, to present on iron complexes in therapeutics.
Thank you, Tanya, and good morning, everyone. My name is Parul, and I'm a research scholar at Panjab University, Department of Chemistry, Chandigarh. I'm delighted to be here to talk about iron complexes in therapeutics. Today, we are going to explore the most amazing molecule that gives literally lives to our blood, that is hemoglobin. This protein plays a central role in maintaining our body's oxygen supply and is involved in several critical diseases as well. Let's begin. So, let's start with the basics. What is hemoglobin? Hemoglobin is a protein found in red blood cells, which is very crucial, and its main job is to act as a transport vehicle carrying oxygen from our lungs to all the tissues and organs throughout our body. After delivering oxygen, it then picks up the carbon dioxide and transports it back to the lungs to be exhaled. The key to this function is iron, which is a core component of hemoglobin and allows it to bind to oxygen. As a fascinating side note, it's this very iron that gives our blood its distinctive red color. These are the key points regarding the hemoglobin. Now coming to the structure of hemoglobin, the creation of hemoglobin is a complex and elegant process. It involves two main parts, the heme group and the globin proteins. The heme part, which contains the iron, is synthesized in the mitochondria and the cystole of immature red blood cells. The globin protein chains are produced by ribosomes in the cystole. These are the complex series of steps involved in the synthesis of hemoglobin. Now, the normal hemoglobin levels varies according to different age. Like in adult males, it ranges from 13.8 to 17.2, while in adult females, it is from 12.1 to 15.1. Similarly, it ranges from 14 to 24 in newborns and 9.5 to 13 in infants. Not all hemoglobin is the same. There are few different types in our bodies. The most prevalent is hemoglobin A, also known as HbA, which makes up over 95% of the hemoglobin in adults. Then there is hemoglobin A2, a minor component at less than 3.5. Finally comes the hemoglobin HbA2, which is minorly present in the adults. It is the primary type found in fetuses, and its level drop to under 2% after the birth. Now what happens when these hemoglobin levels fall too low? This leads to a condition called anemia. So what is anemia? Anemia occurs when your blood has a fewer red blood cells than normal, or when the red blood cells doesn't enough hemoglobin to adequately carry oxygen. The result is often feeling fatigued, weak, and short of breath. There are several potential causes of low hemoglobin. The most common cause is iron deficiency. A lack of essential vitamins, particularly vitamin B12 and folate, can also interfere with red blood cell production. The more serious conditions include aplastic anemia, where the bone marrow doesn't produce enough blood cells, and hemolytic anemia, where RBCs are destroyed faster than they can be made. Also, chronic diseases like kidney disease can also contribute to low hemoglobin levels. So, how to maintain healthy hemoglobin levels? Fortunately, there are many ways to support healthy hemoglobin levels through our diet. Firstly, eat iron-rich foods, which includes animal as well as plant sources. The animal sources includes green leafy vegetables, legumes, and many more. Secondly, boost iron absorption. You can enhance your body's ability to absorb iron from the plant sources by pairing them with food rich in vitamin C like citrus fruits and peppers. Next is get enough folate and B12. These vitamins are vital for producing RBCs. At last, limiting inhibitors can also help in boosting the hemoglobin level in our body. So, beyond low levels, there are also a number of genetic diseases related to the structure and production of hemoglobin itself. The main ones are sickle cell disease, thalassemia, hemoglobin C disease, hemoglobin SC disease. Now, let's briefly touch on what these are one by one. Firstly is sickle cell disease. In this disorder, RBCs become rigid and sickle-shaped, which impairs their ability to carry oxygen and can cause blockages in blood vessels. Next one is thalassemia. Thalassemia is a group of disorders where the body produces an insufficient amount of hemoglobin, leading to different forms of anemia. Next is hemoglobin C disease. This is an inherited condition where abnormal hemoglobin C is produced instead of normal hemoglobin, resulted in chronic anemia. Next is hemoglobin SC disease. This is a type of sickle cell disease that occurs when a person inherits one hemoglobin S gene and one hemoglobin C gene. There are also other hemoglobin variants, including unstable hemoglobin mutants. So, we have talked about low hemoglobin, but can you have too much also? The answer is yes, and it can also be a problem. A condition known as polycythemia is marked by an excess of RBCs. This surplus can thicken the blood and slow down its flow, which increases the risk of serious complications like blood clot, organ damage, and even stroke. Now, these are the iron distribution in different parts of our body. The hemoglobin contains majorly parts of iron, that is approximately 60%. Now I would like to introduce another key protein, that is ferritin. Ferritin is a universal protein whose primary role is to store iron and release it in a controlled manner. While it doesn't carry oxygen itself, ferritin is crucial for hemoglobin production because it acts as the body's iron reservoir, ensuring that iron is available when needed to make hemoglobin. Now coming to a common point of confusion is between ferritin and hemoglobin. Ferritin is a storage form of iron, while hemoglobin is the active working form of iron that carries oxygen. Both are related but serve different roles in the body. Now, while both are proteins involving iron, they are quite different. Hemoglobin is composed of 4 amino acid chains. The powerful example of hemoglobin structure is sickle cell disease, where a change in a single amino acid in the beta chain from glutamic acid to valine is the cause of entire disorder. Ferritin, in contrast, is a much larger complex made of 24 subunits. Now, given the serious nature of these disorders, there are different stages of iron deficiency. First one is reduced ferritin. When it is catched early, it can be cured properly. If ferritin is reduced in our body, the iron storing unit becomes less. Then stage 2 occurs, which is iron deficiency. Next is anemia. But there are standard treatments include blood transfusion and folic acid supplements. Iron chelation therapy is often necessary to remove the excess iron that can build up the frequent transfusions. Medications like hydroxyurea are best to treat sickle cell anemia by reducing the sickling of red blood cells. More advanced options include stem cell transplants to replace abnormal blood cells with healthy ones, and gene therapy, which aims to correct the root genetic cause. All these leads us to an exciting era of therapeutic research, a class of compounds called triazoles. Triazoles are heterocyclic compounds that are widely known for their antifungal properties in both agriculture and medicine field. More recently, scientists have been exploring their fascinating interaction with the hemoglobin. These are the various uses of triazole when binded with the metal ion, that is antimicrobial, anti-ecoplastic, anti-HIV, anti-ulcer, enzyme inhibitor, and many more. So what is this interaction we are talking about? Triazoles can influence hemoglobin's affinity for oxygen. Specifically, certain triazole compounds have been demonstrated to increase the affinity of hemoglobin for oxygen, causing the hemoglobin to bind oxygen more readily. So how do they do this? Let's discuss the mechanism of action. Triazole function as allosteric modifiers. This means they bind to a site on the hemoglobin molecule that is separate from the oxygen binding site. As it is shown in previous figure, the iron binding site is different from the heme protein. Now, a key mechanism involves the triazole compound forming a covalent bond with a specific cysteine residue on the beta globin chains of hemoglobin. This binding action stabilizes the R state or the relaxed state, which is high oxygen affinity state of hemoglobin. By locking it in this state, it prevents the formation of interactions that are characteristic of the T state, which is the tense low oxygen affinity state. Now, coming to the mechanism of stabilizing the R state. Normally, hemoglobin switches between two states. The first one is the T state, which is tense low oxygen affinity. The second one is the R state, which is relaxed high oxygen affinity state. Triazoles help hemoglobin stay in the R state, meaning it binds oxygen more tightly. This is especially helpful in sickle cell disease. Triazoles also influence something called the Bohr effect. This is how pH changes affect oxygen binding. By tweaking this effect, triazoles fine-tune how hemoglobin delivers oxygen in tissues, which is another power therapeutic angle. To wrap up the triazole part, by increasing hemoglobin's oxygen affinity, triazoles keep more hemoglobin in the oxygenated state. This prevents the sickling of red blood cells, which is the root cause of many complications in the sickle cell disease. It's a promising approach in ongoing research. This mechanism is highly relevant for treating sickle cell disease. Remember, in this disease, it is the deoxygenated hemoglobin that polymerizes and causes the RBCs to deform into sickle shape. By increasing hemoglobin's oxygen affinity, triazole compounds ensure and more hemoglobin remains in the oxygenated state. This in turn inhibits the polymerization process and reduces the sickling of red blood cells. Now, to conclude, hemoglobin is much more than a pigment. It's a vital, finely tuned protein central to life, health, and diseases. From genetic disorders like sickle cell and thalassemia to cutting edge therapies using triazoles, hemoglobin continues to be a focus of deep scientific research. Thank you. Thank you all for your time and attention.
Thank you so much, Ms. Parul, for the wonderful session. And now, ladies and gentlemen, we have reached the valedictory session of the GIAN course on Advances in Experimental Therapeutics. This marks the formal conclusion of our academic journey, and we take this moment to reflect on our learnings, express our gratitude, and celebrate the contributions of everyone involved. We would now like to invite a few participants to share their experience and feedback about the course. Um, may I request anyone from the attendees to come forward? Miss Monica.
Good afternoon, everyone. I would like to extend my sincere thanks to Professor S.V. Malhotra for providing deep insights on experimental therapeutics. The lecture laid a strong foundation for understanding the significance and scope of experimental therapeutics in modern medicines. It was a valuable learning experience. I want to thank local coordinator Professor G.J.P. Singh and Department of Chemistry for organizing this workshop. Thank you so much.
Thank you so much, Miss Monica. Next, I would like to call Miss Shreya.
Good afternoon, everyone. Over the past days, we have gone through a journey where chemistry meets biology with the promise of transforming lives through innovative therapeutics. I would like to thank Professor Sanjay V. Malhotra for his insightful lecture on experimental therapeutics. I found it very informative and interesting how chemistry is related to biological aspect of drug development. It has also inspired me to think creatively about solving real-world biological challenges through chemical innovation. Also, thanks to Dr. Aman Bhalla for the wonderful lectures and taking us deep into the asymmetric synthesis and drug development. I would also like to extend my heartfelt thanks to Professor Gurjaspreet Singh, our dedicated local coordinator, whose efforts made this program possible and seamless. Lastly, I would like to thank the National Coordinator of GIAN and the Department of Chemistry for the support in organizing this valuable initiative under the GIAN scheme. Thank you.
Next up, we have Miss Hirsch for the feedback.
Good afternoon, everyone. My name is Harshbir Kaur, and I would like to share my feedback on the lecture titled Advances in Experimental Therapeutics. As we know, Professor Malhotra clearly explained how the discovery of how drug is developed, and he clearly explained how to identify a target, how to, a drug is discovered, and we learned about pharmacokinetics. That is how a drug is absorbed, how it is redistributed, how it is metabolized, and how it is excreted. Also, we got to know the importance for testing of toxicity. I would like to thank Professor Gurjaspreet Singh, local coordinator GIAN, national coordinator GIAN, and Department of Chemistry, Panjab University, for organizing such a wonderful learning experience.
Thank you so much, ma'am. Next, I would like to call Miss Madhu.
Yeah, I'm very thankful to Dr. Professor Malhotra for showing us the role of chemistry in biology, especially in the field of pharmaceutics, and showing us, introducing to us the new techniques in cell and molecular biology. And I would also like to thank Dr. Professor Palla on telling us about asymmetric synthesis, and the research scholars introducing us to new fields and research topics in the field of innovations in therapeutics. Thank you.
Next is our research scholar, Mr. Vinay.
गुड आफ्टरनून एवरीवन। संजय सर से मैंने जैसे कैंसर के बारे में 5 दिन का जो लेक्चर चला, बहुत सारी चीज़ें जानने को मिली हैं। उनमें सबसे ज़्यादा मुझे लगा मोलिकुलर इमेजिंग। मोलिकुलर इमेजिंग में हम क्या करते हैं कि बिना ड्रग के ही हम किसी भी पेशेंट को किसी भी पेशेंट को बिना किसी ड्रग को दिए बस हम उसकी इमेजिंग करते हैं, जैसे रैट्स में या किसी भी एनिमल में। तो उससे क्या है, उस चीज़ को हम केमिस्ट्री में भी लिंक करवाया उन्होंने कि जैसे हम केमिस्ट्री में डिफरेंट आइसोटोप्स पढ़ते हैं, उन आइसोटोप्स की हाफ लाइफ क्या है और उसको हम एज अ ड्रग डिलीवरी या इंटरेक्शन, ड्रग कहाँ जाके इंटरेक्ट करेगी, वो सारी चीज़ें हमें पता चली है। और साथ में मेरे को सबसे अच्छा एक और लगा कि हम एंटीबॉडीज हैं, एंटीबॉडी हमारी बॉडी में है, वो क्या करती हैं, किसी भी डिजीज के अगेंस्ट फाइट करती हैं। हमने क्या किया कि एंटीबॉडीज के साथ जो भी हमने ड्रग है उसको अटैच करवाया। उससे क्या हुआ, एक तो एंटीबॉडी अपना इफेक्ट दे रही है, साथ में उसके इफेक्ट को एनहेंस करने के लिए हमने उसको ड्रग के साथ कोऑर्डिनेट करवाया था। तो ये सब चीज़ें और भी बहुत सारी चीज़ें हैं जो मुझे इस 5 दिन की लेक्चर सीरीज़ के जानने को मिली है। आखिरी में मैं लोकल ज्ञान कोऑर्डिनेटर प्रोफेसर गुरजसप्रीत सिंह का धन्यवाद करता हूँ। थैंक यू।
Next up is Miss Rashmi.
Good afternoon, everyone. I would like to take a moment to share my reflections on the lecture Molecular Imaging in Drug Development. The lecture was highly insightful as it focused on the journey of small molecule drug discovery and development. The session explored high throughput screening workflows. The case study on anti-cancer small molecules stood out for me. It demonstrated how scientists balance potency and toxicity and use biomarkers to select patients population most likely to respond. This practical application made the concept very relatable. We discussed preclinical testing and translational models, pointing out the challenges of predicting human outcomes from animal studies and the importance of multidisciplinary collaborations to move therapeutic candidates closer to clinical trials. This segment of session provided a comprehensive overview of the drug development pipeline, and it underscored the complexity and teamwork required to bring a molecule from lab bench to bedside. At last, I would like to extend my sincere thanks to local coordinator of GIAN, Professor Gurjaspreet Singh sir, and Department of Chemistry for their support and encouragement throughout this journey. Thank you.
These were some wonderful feedbacks from our research scholars. Now I would like to invite someone from MSc interns. Okay. Ms. Renuka will come forward.
Ladies and gentlemen, respected teachers and esteemed guests and my fellow graduates, it is an incredible honor to stand before you today as the valediction of the seminar. I thank to all the coordinators, organizers, Department of Chemistry, Panjab University and the international speaker, and especially to Dr. Aman Bhalla to taught us about the asymmetric synthesis in a very fascinating way, which has a very wide application in pharmaceuticals, agrochemicals as a biological activity and creating novel materials. Thank you everyone for being present here and to make this event successful. May we continue to learn, grow and achieve greatness. Thank you.
Next up we have Miss Anisha.
Good afternoon, everyone. I would like to sincerely thank our local coordinator, Professor Gurjaspreet Singh, and the course coordinator, Dr. Bhalla, who took the initiative to organize this 5-day academic program under the GIAN scheme here at Department of Chemistry, Panjab University. Your effort and coordination have brought us to rare and valuable opportunity, and we are genuinely grateful. This seminar offered interdisciplinary value that helped us on how chemistry and biology must work together hand to hand, from understanding molecular interactions to optimizing drug formulation and delivery mechanism. We hope this seminar marks not the end but the beginning of a lasting academic collaboration. Thank you, sir, for making this experience wonderful and truly unforgettable. Thank you.
Next, I would like to call Ms. Muskan.
Good afternoon, esteemed organizers and participants. It is with great respect and sincere appreciation that I take this opportunity to thank our host institution and in particular Professor Sanjay V. Malhotra, the local coordinator, Professor Gurjaspreet, and the coordinator, Dr. Aman Bhalla, Department of Chemistry, Panjab University, for the exceptional effort in organizing this GIAN workshop on advancement in experimental therapeutics. This gathering has been both intellectually stimulating and professionally rewarding. The depth of knowledge shared, the range emerging therapeutic strategies presented, and the caliber discussion reflect the significance and urgency for advancing and our understanding in this evolving field. The interdisciplinary perspective presented here have reinforced the importance of collaboration between basic scientists, clinical researchers, and industry partners in accelerating the development of safe and effective therapies. Once again, I extend my sincere thanks to organizers, speakers, and all participants for making this workshop a remarkable and enriching experience. Thank you.
Lastly, we have Ms. Sanchi for her feedback.
Good afternoon, everyone. I am truly grateful for the opportunity to participate in this GIAN course on academics in experimental therapeutics at Panjab University. A heartfelt thank you for the GIAN course should express gratitude for the opportunity, acknowledge the contribution of the faculty and organizers, and highlight the value gained from the experience. Thank you.
Thank you everyone for your heartfelt reflections. We are glad to know that this course has left a lasting impact. We will now proceed with the distribution of participation certificates. I request our local coordinator and Professor Gurjaspreet Singh to kindly join us at the front. To begin with, I would like to call Ms. Nishu. Next up, Ms. Hina. Ms. Parul. Ms. Hema. Ms. Priyanka would be next. Next, Ms. Ritu. Deepika. रश्मि नहीं दिख रही ना. Miss Rashmi Bhardwaj. Next up is Mr. Anurag. Miss Renuka. Anshul Janeja. Archita Sharma. Harshbir Kaur. Muskan. Cheshtha. Muskan. Miss Swati. Miss Sanchi. Miss Lavisha. Miss Poonam. Musabbar. Shubha Matwal. Anisha. Mr. Vineet. Miss Madhupreet Kaur. Mr. Manish. Miss Anshu. Neha Sharma. Mr. Vinay Kaushik. Ayushi Sharma. राजन का सर्टिफिकेट कहाँ है? अंकुश सैनी. महक. तनिषा. सुमेश खुराना. रात से फोन नहीं लगा. ठीक है. कहाँ है ये तो आई नहीं है. मिस्टर मिथुन. इसी को यही दे देंगे. ये है नहीं. मिस्टर मनराज. अरे वो राजन सिंगला का पूछ के आओ. Miss Monica. Miss Shivani. हाँ यही दे यही तो मैं बार बार पूछ रही थी कहाँ है कोई नहीं चली गई चली गई राजन शर्मा चल जा अब तू एक मिनट मेरा नाम कौन बोलेगा मेरा रहने देता नहीं श्रेया ठाकुर मेरा नाम लेकर खुद ही चली जाती है
Miss Tanya Dheer.
We have special appreciation for our speakers. I would like to call Miss Parul, Miss Priyanka. Miss Hina, Miss Poonam, lastly Miss Renuka. यार वो लड़की का नाम पूछ एक और रह गई है वाइट शर्ट वाली मिस तरनप्रीत कौर सॉरी मनरीत कौर नहीं बस हो गया As we draw the curtains on this enriching academic experience, we express our deepest gratitude to the Ministry of Education, Government of India for supporting this initiative under the GIAN scheme and to Panjab University Chandigarh, Department of Chemistry for providing all the institutional support. I would like to take a moment to especially acknowledge our dedicated course coordinators, Professor Gurjasprit Singh, Local Coordinator, Department of Chemistry, Panjab University, Dr. Aman Bhalla, Course Coordinator, Department of Chemistry, Panjab University. Your efforts, vision and guidance have shaped this course into a truly impactful learning experience. I thank all our resource persons, coordinators, organizing team and participants once again. We hope this course has not only added to your knowledge, but also sparked new ideas and collaboration. Lastly, I would like to invite Professor Gurjasprit Singh, Course Coordinator, for the welcome address, for the valedictory speech.
So, thank you, Tanya. So, very good afternoon to everyone. As we come to the close of 5-day workshop on advances in experimental therapeutics, it is time to reflect on the enriching experience we have shared and to extend our heartfelt gratitude to all who made this event a grand success. First and foremost, I would like to thank the worthy Vice Chancellor, Professor Univ of Panjab University, Chandigarh, Dr. Nil Kumar, the National Coordinator of GIAN from IIT Hyderabad, Professor Gangaram for their invaluable support and encouragement. I also extend my sincere appreciation to Professor Sanjay V. Malhotra for gracious accepting our invitation joining us for this workshop and generously sharing his expertise and insights with all the participants. Our special words of thanks goes to course coordinator Dr. Aman Balla for its unwavering dedication and tireless effort in planning and executing this workshop seamlessly. I would also like to express my gratitude to Professor Narinder Singh, Dr. Dharamveer, and our core organizing team, Hina, Pallavi, Poonam, Parul, Pooja, Priyanka, and Renuka, for their meticulously planning and coordination behind the success. A big thanks to Somesh, Manraj, Mithun, Vineet, Tanya, Shreya, Monica, Shivani, and all the participants who contributed with great energy and enthusiasm to this session. I sincerely hope that each one of you had a meaningful and insightful learning experience. I am confident that you are living with valuable knowledge, new ideas, and renewed passion for research in experimental therapeutics. Thank you once again, and I wish you all the very best in your future endeavors. Thank you.